First of all, thank you for letting me the opportunity to present our work. My name is Alejandro Serrano Sa from the Minimal Cell Biophysics Group at the Centro Nacional de Tecnología. I'm currently doing my PhD with James Pelletier, and I'm going to present you today our work that is titled The Effect of Cell Division-Related Genes on Cellular Growth and Size on the Minimal Cell JCBI SYN3A. First of all, I want to introduce the team and the people that has collaborated with us, starting with the people from the Minimal Cell Biophysics Group, which would be James Pelletier, Marina de la Fuente, and Pepe Blasco. Also, our collaborators from the Centro Nacional de Biotecnología, Cellular Group, our collaborators from the CIB at Madrid, Germán Rivas and Silvia Zorrillas Group, especially Diego Laxalde. Also, Begoña Monterroso's group from the IQF at Madrid. And finally, John Glass from the JCBI. First of all, I want to start putting the focus on our research in the cell division related genes, specifically those of the division and cell wall cluster that is present in the minimal cell strain JCBI SYN3A. And this is important because the minimal cell provides the perfect simplified model to study these genes, how they interact and what effects do they have both in cell divisions and the fitness of the cell. From all these genes, we're going to focus more on the named cluster 1 genes, that is FTSZ, CPF, and the gene 520. Of genes, the most characterized one is the FTSZ, which is a highly conserved protein that produces a cytoskeletal protein. It also has the property of being a GTPase hydrolase. It can form dynamic filaments that normally interact with CPF and FTSA to interact with the membrane and be involved in the cellular division processes. And another thing that makes FTSZ interesting for our research is previous studies from Moger and Reicher lab, in which we didn't took part, that shown that one single point mutation in the sequence of FTSZ can lead to a huge gain in the fitness of the cell. And when this mutation was translated into the minimal cell, it was alone that mutation responsible for a gain on its fitness. With that being said, what we want to really study is the differences in the cell division phenotypes across different minimal cell strains, because normally we used to categorize cell division phenotypes into two big groups. One will be the strains that appear as small cells, similar to the wild type, and the other will be those strains that appear to have large forms or big objects that we can't really know if they are cells. For example, JCBI SYN3A, which has FTSZ, and JCBI SYN3.0, which doesn't have FTSZ. The thing is that when you add the cluster genes 1, 2, and 6, which include FTSZ to JCBI SYN3.0, it recovers the original or the small cell phenotype, but deleting FTSZ from this cluster brings it back to the large form phenotype. The thing starts to get complicated when we took JCBI SYN3A, which has FTSZ, and delete the cluster 1 genes, including FTSZ, and the small cell phenotype is not lost. It still remains as small cells. But if we only add FTSZ to the JCBI SYN3.0, it doesn't get the small cell phenotype. It still remains and appears as large objects and large forms. This raises a natural question that what is the real effect of FTSZ in the fitness of this cell, and what effect does it have on the cell growth and the cellular size? To address this question, we are going to be comparing these two strains that will be the JCBI SYN3.8 that has FTSZ and the JCBI SYN3A that lacks the gene cluster 1, including FTSZ. First of all, I'm gonna show the comparison of the growths. To estimate the growth or the doubling time of these strains, we measured the doubling times in a plate reader using the Signal 650 nanometers wavelength to achieve these curves. And by fitting these curves, we can obtain the doubling times of these strains. Another method to obtaining the doubling time will be the making serial.
Okay.
It's everything's fine?
Yes.
Okay. Another method for estimating the doubling time will be to make serial dilutions of these strains and measuring the amount of time that it gets for the 2x dilution to achieve the same optical density value by calculating the lag time, the delay time between both curves. By combining these two methods, we can estimate some preliminary results on the doubling time on both strains, with some results that show that the deletion of cluster 1 significantly decreases the doubling time, meaning that the JCBI SYN3A that lacks FTSZ and the other two genes of the gene cluster 1 grows faster, with an estimated doubling time of 2.8 hours versus the 3.5 hours of the wild-type JCBI SYN3A strain. And sorry, this could be because of several reasons, but two ideas that we have is that the expression of FTSZ may be causing an excessive cost for the cell that slows down its growth, and also the inner activity of the FTSZ, because it's a GTPase, will be draining energy from the cell and slowing down the cellular growth. Regarding the expression or the metabolic burden that can be caused because of the expression of FTSZ, to deepen into this question, we engineered inducible promoter systems regulated with the TET-R sequence and using the lactate dehydrogenase promoter to overexpress a protein that doesn't have the GTPase activity to see what effect does it has in the cellular growth. To do so, we choose the lactate dehydrogenase promoter because it's one of the most expressed proteins in the JCBI SYN3A proteome, and we added the TET-R and some small changes to optimize the system and make it an inducible and very controllable expression system to see the effect of the metabolic burden. And for the activity of the FTSZ, the GTPase activity, our collaborator from the CIB, specifically Diego Laxalde, has estimated the GTP hydrolysis activity of FTSZ by characterizing FTSZ in vitro. As we all know, FTSZ is able to hydrolyze GTP into GTP and phosphate, and using the malachite green assay, they were able to measure the phosphate that is liberated into the media, measuring its absorbance at 630 nanometers, and they measure it every 30 seconds for 2 minutes assays, with a final result of 3.5 micromolar of phosphate liberated to the media per minute and per micromolar of FTSZ. Additionally to this, we wanted to see the effect of FTSZ in the size of JCBI SYN3A, because we have seen that it grows without FTSZ. It seems to grow faster, but we wanted to know if it was causing any abnormal size or shape into the cell. To do so, we developed a protocol to use cells in culture, mix them with molten agarose to immobilize them, and then tau-stet microscopy to achieve a high resolution images with using the membrane dye FM464. And as you can see, we can achieve a single cell accuracy on the images with one value of pixel of several orders of magnitude less than a micrometer. We also checked the general shape of the populations of the culture with no big difference between the strain that has FTSZ and the one that lacks the gene cluster with FTSZ. And we also measured the average diameter of these cells to make sure that the absence of FTSZ wasn't causing any abnormal sizes, nor bigger nor smaller ones. And we found no significant difference between both of them, with diameters ranging from 0.4, 0.5 micrometers up to 1.2 micrometers in diameter for both strains. And with this, I want to end the talk, giving value to two main points. One would be that the metabolic cost that FTSZ may be causing to a cell, because we know that both the translation and the GTP hydrolysis has a cost for the cell that may explain this faster growth when FTSZ is not present. And this raises the question that there should be another mechanism from the cell division that is not the cytoskeletal protein FTSZ, such as the crowding of membrane lipoproteins or the membrane curvature. With this, I want to say thank you again to all our collaborators and other people that has helped us in this research and our funding, and I will be happy to answer questions.
Lovely, very, very nice. So, questions for Alejandro? I see you've got applause. Ingguang, the floor is yours.
Very nice talk. Yeah, just a quick question. Like, you reported the doubling time of GCVI like as 6.5 hours. Is this because of the different like methodology you use? Because we think the doubling time is roughly like 2 hours, right?
Yeah, we think that we know that it is different from some doubling time estimations of other publications and other groups. And we think that it is probably due to the conditions of the growth that may have some small changes between one experiment here in our group and other replicates in other places. But we have repeated this a couple of times, and the doubling times are consistent. So, yeah.
Got you.
I'm surprised at the difference in the growth rate that you measure and we have measured, Alejandro. And I'm wondering if there are just really subtle things we do differently in terms of how we make our SP4. I don't think that we talk about a slower, gentler life in Spain, but I don't think that has to do with it.
Yeah, we think that's probably the reason for it. Some small changes in the media or something like that on the general procedure that may be causing the difference.
On, you're up. Got a question?
Yeah, just a small thing. I think so last week we just had a preprint come out where we see the cell size actually changes when we switch the lipid diet in the minimal cell. So it's for me really fascinating to see that the cell size doesn't change as much when you knock out FTSZ. So maybe I can send you the paper later and maybe we can keep it in discussion.
Yeah, it would be great.
I will note that we found that having really high level expression of nchez in our hands. So Kim Wise showed a 10-15% difference in growth rate that was reproducible.
Mm-hmm. Yeah, we want to try something similar with the inducible promoter system.
Okay, moving on.
There was one question in the chat, John.
Oh, I didn't see it.
The question is, could be variability of growth medium to perhaps serum variation? And yeah, I think the growth media will be a high factor on this difference. And as we don't know the exact composition of the serum, that may be a huge factor.
So you're using fetal bovine serum as opposed to the knockout that we use?
We use the knockout.
Okay, all right. Well, that you would think that would be consistent still. Your data looks precise and reproducible, and different places, different things.